Okay. So yeah, I'd like to welcome you all to our seminar series for today. Just a little bit about myself. I'm Perry Acree. You may notice that I am not Beth. She's a little under the weather today, so I'm filling in. But yeah, I'm Perry Acree. I'm a visiting scientific specialist for environmental contamination at the Illinois Sustainable Technology Center. And just a little land acknowledgement for today. As a land grant institution, the University of Illinois at Urbana-Champaign has a responsibility to acknowledge the historical context in which it exists. In order to remind ourselves and our community, we will begin this event with the following statement. We are currently on the lands of the Peoria, Kaskaskia, Piankashaw, Wea, Miami, Mascoutin, Odawa, Sauk, Muskogee, Kickapoo, Potawatomi, Ojibwe, and Chickasaw nations. It is necessary for us to acknowledge these Native nations and for us to work with them as we move forward as an institution. Over the next 150 years, we will be a vibrant community inclusive of all our differences with Native people at the core of our efforts. And a little bit about the webinar series. This webinar and all the Illinois Sustainable Technology Center's webinars are certified green events through the University of Illinois Institute for Sustainability, Energy, and Environment. To find out more about certified green events through U of I, please visit sustainability.illinois.edu. To find out more about the Illinois Sustainable Technology Center's webinars or to sign up for these events email list, please visit istc.illinois.edu/events. A couple of housekeeping items. Today's webinar is being recorded. The recording and the slides will be available for viewing online within about a week, and an email will be sent when that is ready. I'd like to ask everybody to please remain muted. Questions will be through the Zoom Q&A feature. You will be able to upvote questions you like within Zoom Q&A using the thumbs up button under the question. I'll be reading the questions to the speaker at the end of the webinar. At that time, the most popular questions will be asked first, followed by questions in the order which they receive. If you have any technical difficulties, please send me a private chat message. Okay, so today's speaker is Zhang. Win is an associate professor in the Department of Civil Engineering, Civil and Environmental Engineering, and Chemical and Material Engineering in the Newark College of Engineering at the New Jersey Institute of Technology. His research focuses on colloidal interfaces and processes that are crucial for environmental and chemical engineering applications. Wynn's research embraces environmental behavior and interfacial processes for nanomaterials, microplastics, and soft particles such as microbes, bubbles, catalytic/reactive membrane filtration systems for desalination, record resource recovery and emerging contaminant removal, photocatalysis, and microalgal removal and harvesting. Wynn earned his PhD from the Georgia Institute of Technology. He is a licensed professional engineer registered in the states of New Jersey and Delaware, and is an American Academy of Environmental Engineers and Scientists board certified environmental engineer. So thank you to Wynn. Yeah, and I will leave you to it.
All right, thank you, Perry, for the kind introduction. Also very pleased to have this opportunity. Are you able to see my screen? Just share my monitor number 4. I have 4 monitors. Just make sure you're able to see my slide, the projected slide.
We can see it.
Okay, cool. Okay, yeah, thank you everybody for joining this webinar. I already seen some people that I know very well. Thank you for your time. Okay, so today I'm going to talk about the challenges that we have for microplastic, particularly nanoplastics, detection and separation, things like that. So as you see from this slides, and basically the challenges all come from the major steps when we analyze the environmental samples, right? So if we do not, deal with the environmental samples, usually those challenges may be, less stressful or, basically gone. But in terms of environmental samples, we are, facing the problem of separations, purifications, so that we can eliminate the interferences, as you can imagine. And then also, usually the plastic particles in the natural, in the environmental matrix are very low, even though they are everywhere, but the concentration abundance are still much, much lower. So that will certainly pose, certain, challenges on detection size, because, all these instruments have detection limits. Okay, I'm gonna move my slides. Oh, it's not moving. Come on. Oh, sorry about this technical difficulty. I'm not sure where my browser, let's see here. Just bear with me, I'm trying to find out my cursor. Okay, right here. But this is not. Maybe I need to exit and then project it one more time. Okay, find one my, okay. So here we go. So first of all, before we getting into the technical discussions about the challenges of detection, I'd like to give people a very, overview of the challenge, the plastic pollution in general and the threat to, our natural system ecosystems. And by, doing so, I try to motivate people to have a compassion into our Earth and also our coexisting species that who are really suffering from this pollution, perhaps more, suffer than we have. So secondly, I'm going to talk about the nanoplastics and their unique properties, because basically the challenges primarily come from the nanometer sizes we're dealing with. For large bulk plastic particles, usually we have much lesser, problems. And then we're going to talk about the major steps of nanoplastic characterization, and then, certainly discuss the challenges associated with the different steps. And then while last, I'm going to stress on a few of innovative technologies out there, but maybe not many people are aware. Okay, so as you see, the plastic pollution has been there for decades, since I was elementary schools, probably middle school stage, we were already educated that the plastics has been widely used in food industries and many industrial processes. And over time, you see that we dump way too much plastic pollution or waste materials in the natural environments. As you can see, this short video shows a river, a creek, that pushing tons of plastic garbages all the way to downstream, perhaps the marine environments. And then, at the end, we are going to be the central point that, eventually get back to us, even though we dump to the environment due to the food chain system, and we are going to actually sacrifice our health. For example, a study that carried out by University of Newcastle and in Australia that published the results in 2019 showing the human feces here, 10 milligram human feces, average would contain 20 plastic particles. And we digest over 2,000 plastic particles per week and per capita. That means you may wonder why and how come we, ingest so much plastic particles. And think about the food that we eat, like a sea salt. The salt contains plastic particles. Even the tap water, drinking water, may potentially carry on some plastic materials, because in drinking water system, drinking water treatment, usually, those processes are not originally designed to treat or remove the plastic particles, and that's especially those small particles. So if you look at this, this is the natural or ecological consequences we're seeing every day. Maybe see this seagull birds, a kind of birds eating too much plastics and eventually died from indigestible plastics that accumulated in the stomach. And even for their little baby birds, they're fed with the plastics from time to time, and that would certainly cause a lot of mortality, higher or increase the mortality rates. So there are certainly other hazards, risks posed to the natural environments. For example, the plastic material could release the toxic substances like plasticizers or estrogenic substances that cause genetic mutations or health problems. As you can see here, once again, my slides is not moving. Sorry about that. I can see my cursor. Okay, here we go. So this little whale is already dead because of feeding on the toxic milk from the mother whale. So it's already dead, but it's due to perhaps the pollution from the marine environments, and particularly for the nanoplastic. Why do we care? So the reason, I guess, everybody is aware that we are imposed to a plastic-laden environments, increasingly, with increasing intensity now. And not merely the bulk plastics that something can see, for example, on this table, you can see the microplastics generally defined as plastic fragments particle that has a diameter less than 5 millimeter. That's something we can see, right? And then we go down to the sizes like a few hundred nanometers, like 200 micrometers. Then those are basically the resolution limits that human eyes can detect. So anything that below 200 micrometers, then we're not able to see. But they're posing quite uncertain risks in terms of health risks and environmental risks. So for example, here, the plastic particle, once they get into certain sizes, that we call them nanoplastics, something in terms, in general definition, nanomaterials or nanoparticles should have a one dimension that is less than 100 nanometers. That's pretty much a common understanding for nanoparticles. So when you look at the other biological substances or biomolecules like DNA viruses, they are typically the size of less than 100 nanometers. They're very small, but also they are pretty diverse in terms of compositions and sizes and shapes, and even their surface and properties. For example, they may be negative charge or positive charge depending on the surface coating, or they could be made of polyethylene, polypropylene, polystyrene, all kinds of polymers, synthetic polymers. They are not supposed to be there in natural environments, but due to our human activities, we overproduce them, and they also we over dump them in the natural environments. And more critically, here I've just put some highlighted words here. So not merely because we're not just looking at the backbone polymers, we also have the problem of chemical additives, such as I just mentioned, plasticizer, stabilizer for flame retardant, antimicrobial reagents, and some other light absorbing agents for colors, or absorbing lights properties. So those chemical additives, even though they might be low, in concentration compared to the backbone polymers, but they pose much higher risks because of their own toxicity and perhaps some of the chemically resistant properties. Also, the copolymers, for example, monomers, dimers could also release as the backbone of polymers, microplastics, or plastic in general, undergoing aging processes. Those copolymers could also pose higher risks. Okay, well, I don't know why my slide's not moving always. Sorry about that. This is quite annoying. Okay, let me see. I have to reproject one more time. I don't know why it used to be working quite well. Couldn't see my browser. Maybe once I share my screen, something wrong with my browser right here. Okay, so maybe I just do this. Instead of projecting my slides, I just show, as long as you can see my slides contents, then it shouldn't be a problem. All right, so you can see that just to summarize really quick to give ideas of how different the nanoplastics are, in terms of their different properties compared to the regular bulk particle that we're able to see. So so in my opinion, certainly they are polymer particles with high polymer contents, and they are insoluble in water and usually persistent in the natural environment. So by saying that, if they are naturally, or they are biodegradable or degradable in general, then they are not going to be a problem, right? And unfortunately, most of them are, at least for a certain period of time, they are chemically inert. They are not really able to degrade over time very fast. And then they also may appear to be aggregated due to their surface affinity, high surface areas, and due to the hydrophobic or or electrostatic attraction, they may be aggregated in aggregated forms or dispersed depending on their surface properties and matrix, the salinity, pH, a lot of water chemistry associated with their stability in water. And because of their small, they usually have very high diffusivity and mobility, which means that they may enter food chain systems in a faster path, faster rates compared to the bulk particles where they're relatively lower in terms of ingestion by animals. And they may accumulate or bioaccumulate similar to the commonly started nanoparticles, and they may even penetrate through certain barriers. For example, the human brain, and people have been quite concerning about the plastic entry or similar to the nanoplastic entry into the brain cells, and because they're so small, they may. They also exhibit quite strong dependence, size and shape dependence of their surface properties. For example, their surface charges could be much higher or greater than their large bulk particles. They also have a certain crystallinities. Some of the polymers have a crystallinities that may affect the light absorbance and also surface reactivity. And I do see the people have been studying the crystallinity changes over time. When the plastic particles undergoing aging processes. So aging again would somehow leach out the secondary pollutant, as I just mentioned. And here I have shown a table that people generally understand. When you analyze their compositions, people tend to use the FTIR, Raman, or other analytical tools, as they're polymers. And then the shape we can use as EM electron microscopes or just regular microscope, fluorescent microscopes, depending on the detection limits. But all of this, instruments are out there, but they're not really, I would say it's not really straightforward to utilize for the plastics particles at small sizes, because again, the challenges or hurdles we're going to go through together today. Just give you a very broad overview. So this is on my left hand side. So it's here. So the outer boundary here, we're looking at the plastic materials. They're undergoing degradation, crushing, or they broke down into small pieces like microplastics and the nanoplastics. And then in order to analyze those environmental plastics, then we need to go through first step is a pre-treatment and separate. Pre-treatment means basically you need to eliminate those coexisting organic matters that could come from the naturally organic matters or proteins, bacteria, anything that are here or deposit on the surface of the plastic. You need to certainly take them out before you analyze them. Also, you need to separate the plastic particles. Usually, that's a very difficult part because think about the concentrations, low concentration as well as the small sizes that are not easy to separate. And then the first characterization will be visualization and basically give us a visual evidence of the plastics. And also there are a few challenges, right? Because they're tiny, and also by using the regular optical microscope, you're not going to really tell the compositions. You don't know what you're really looking at. So there are lots of additional characterization steps we need to undertake. And ultimately, the goal is to quantify, right? So in terms of water treatment, we really need to know how much we're dealing with and how many particles in water. And then after certain treatment processes like the coagulation, filtration, you need to quantify how many residuals, not merely to look at whether we have or we do not have. We also need to know the treatment removal efficiency. So on the right-hand side, this is a paper, table of contents from a publication that's showing the complicated steps for the plastic particles of extraction and analysis from the biological matrix, for example, fish, mussels, plankton. So or wastewater that contains numerous of coexisting pollutants. As I just mentioned, you can certainly use different type of particle capture mechanisms such as filtration or centrifugation or other means. But due to the sizes, nanoplastics, usually that's not really easy to achieve. And then even though you separate them, maybe you're not able to analyze them very well because of the, again, the concentration or small sizes. You have to use specialized instruments like, ICM, electron microscope, or special Raman IR to increase your resolution power. Otherwise, again, you're not able to confirm the identity of those polymers. So again, the conclusion here I said is there really no universally accepted protocol. I would say there's really no, just similar to nanoparticles, there are lots of problems with the standardized protocol to detect the nanoplastics in the natural environment, in the environmental samples. So here again, the context is about the environmental samples. So you're dealing with studies that involve the use of a standard plastic beads or particle, then you do not have these challenges again. Okay, so I'll start with the pre-treatment and then just going through all the major steps. So the pre-treatment is a step that we usually use chemicals, chemical cleansing to cleanse up, the plastic particle, their matrix using the solvents, organic solvents, or like ethanol, methanol, or acid base to dissolve the salt or any dirts, inorganic matters, and some organic matters, so that we can largely prevent the interferences from those coexisting pollutants. But the problem is, as you can see already put down here, some of the plastics are vulnerable or fragile. They may undergo a mechanical or chemical degradation because, for example, you use the solvents and cleaning plus a sonication. And sonication is known to produce cavitation-based radicals, and also such high-frequency mechanical vibration would somehow decompose. You see here, it's really just a mechanical decomposition. And also the acid base, usually the plastics are not really able to tolerate high alkalinity. For example, the plastics, so-called polyethylene terephthalate plastic PET could quickly undergo a degradation when it's immersed or exposed to sodium hydroxide. This is a well-known degradation pathway, so that when you deal with such plastics, PET, which is commonly used as our bottled water, this is usually used PET. So be careful when you deal with such plastics, and just make sure they're not going to be degraded or, when you pre-treat them. And the separation is also very difficult. For example, the red box here basically capture the major methods people have been using. For example, using the nets, and Newton nets or filters to capture the large pieces of plastic fragments. But those methods or filters are not really suggested for nanoplastics because of their small sizes could allow them to penetrate the filters very easily, and they're not going to be captured. And even though they're captured, you probably know that there are a lot of, for the filtration, membrane filtration, there are a lot of material that actually here, this table shows you the membrane filters are made of polymers, right? They could made of polymer sulfones, PEs, Ca, polyamides. Those are basically polymer fragments or microplastics that we are dealing with. So it's not a good idea to use those organic polymer-based filters, because those filters will generate quite strong signals when you analyze them under Raman or FTR. So here I want to highlight a recent study that's basically showcase a combination of nanotechnology with surface enhanced Raman spectrometry. So instead of using the traditional or commercial filters, people have done this. This group has utilized a silver nanowire to basically fabricate a layer of a silver nanowire filter. So with that filter, they use this filter to filter the water sample containing plastic particles, and then utilizing the filter without the dumping or without separating the plastics from the filter, just directly use that filter and for the Raman mapping, which is a very nice idea because you have a silver to enhance the Raman signal. In the meantime, you do not have the trouble to separate the captured plastics from the filter, which is very, very painful, right? You know, especially the nanoplastics are very low in numbers. It's not easy. You're not able to visualize the plastics, so you never know whether you can take them out from the filter. So this gives a better idea to shape, our future research. And also you may consider the use of inorganic filters right here using the, aluminum oxides, some other composite metallic filters to lower the background noise from the polymer matrix. Another separation techniques here besides the filtration, and this, I have no practical experience, but I've been hearing this for decades. When we deal with the nanoparticles in general, people have been using so-called flow fractionation, where you basically have a tubing, a very, very small tubing, and you can apply different kinds of external forces to separate out the particles of interest. For example, you can apply the very famous one is the centrifugation, right? You can use a different centrifugation G-force to settle down the heavy or high density particles, and then you can separate out those from the matrix, or using pressure or using electrical fields to again to rely on the surface charge of the plastics, and also for those ferromagnetic particles, you can use the magnetic fields. But again, as you see this, the principles of a separation, you probably understand that there are a lot of problems for plastic particles, right? So some of them might be low in density or high. Some of them are, neutrally charged or negative charged. They're very, diverse in terms of their surface property. They really know such, standard methods to, to separate out the particles. First of all, you need to understand, perhaps what they're out there in the sample first, and then you make certain hypotheses and driving the separation using their hypothetical properties, and then you need to validate. But even though there are lots of uncertainties, and this technique has been widely utilized, see here, it's pretty powerful in terms of separation. They could separate out the plastic particle less than 1 micrometer, and even for PS and PE particle, the people have done pretty well separation for the nanoplastics. You can see ranging from 50 nanometers all the way to 1,000 nanometers. And then once you separate out, let's say you separate out the plastics, you're sure that there are some plastic particles on your filter or on whatever, liquid fluids, then you're going to perform a visualization step to visualize them, right? You either use, usually people tend to use first, optical microscope. If they're large, then you can certainly see the plastics, and if they have a color, then it will help you differentiate them from the matrix. And or if the plastics are able to be labeled with a fluorescent dye, or they are fluorescent plastic particles, then they can use a fluorescent microscope like a confocal. You can really, quantify and also look into different depth of samples. And lots of studies employ the electron microscope as a routine. So they can utilize, the morphological analysis from the electron microscope. Moreover, they could see the elements, distributions, but primarily the carbon and what else? Carbon and some other, elements that are associated with the plastics. But the problem is for SEM and plus EDS, the problem is, if you understand the principle here, there's lots of, I guess the chemists are not, really paying attention to this limitation. So if we're dealing with the small size particles, and you want to look at the particle, differentiation, right? But the problem with the SEM EDS is that the principle that would not allow you to look into the compositional information in such detail or in such small resolution, because if you look at this graph here to show you the electron beam shooting at the sample surface, right? The sample surface within certain depth. maybe a couple of, usually a couple of micrometers in depth. And then, we rely on the scattered electrons to detect the sample information, morphological or X-ray. The X-ray will come from, the deeper side. But again, so this the sample region where the electrons is scattered is huge compared to how huge, as I just mentioned. The resolution, resolving power is really just, a few micrometers in diameter. There's a sphere, the spherical region of the sample would be entirely bombarded by the electrons, basically. So you're going to basically review the composition information through the EDS for the entire region of the sample, not just the surface of the, let's say, a few nanoparticle. You cannot resolve such, information yet. Also, for TEM or SEM, you need to have a few, sample preparations, right? You need to load the samples on, and you have a certain exposure to, to the air. At least you may have a, cross contamination. That probably this is a common problem, not merely for SEM, TEM. But beyond that, I also listed some others, some other tools like AFM, and the other one is dark field hyperspectral imaging, and I'm going to talk about that soon. So here I want to show you a recent study that just published, and it shows the combination of ICEM and Raman. So this is a fantastic idea. I've been thinking about that, but again, I'm not a physical chemist. I just know the principle. It is quite doable. Now people have achieved this combination pretty well. So basically, you see here, this is a regular Raman microscope. You can see the things, but not really, having a good resolution. And then with the ICEM, because you're using electron beam as, the scattering signal, then you can have pretty good resolution. But the problem is, again, when you look at these particles, you never know what they are, right? They could be just, interferences, not really the plastic particles. If they are environmental samples, right? If they are not, they are, synthetic, then you don't need to worry about that. And then with the Raman signal, I guess it is concurrently operated, then you can generate the Raman signal or Raman mapping that would help you differentiate. For example, based on the signal intensity or the Raman signal, you can see the color differences from the the backgrounds, as well as the sample, the large piece of the sample, which is aggregated polymer PE particles. So this is a very good combination of hybridized system. And then in terms of dark field hyperspectral imaging, I have tried before pandemic, I tried to link up with a few of my colleagues who own these instruments, and I said this is could be a potential tool for nanoplastic, not merely for the nano silver, nano gold, because from the company, they usually post an advertisement of a nano silver, nano gold. And they use this for imaging, as well as characterization of the particles number and states, surface states. So, but unfortunately, the pandemic is slowing down our research progress. We're not able to really get good results. But to my limited, understanding of this technique, it is not easy, even though the results tend to be fancy. It doesn't require electron beam, right? It's just using a regular optical light sources, and but then you have a special numerical aperture design or something to enable the maximum or the increased absorption or scattering of the lights, so that the materials, different materials would scatter different lights at beams of different wavelength and certain frequency or certain intensity based on the color, very minor color difference between the identified particles. You're able to tell the compositions or surface compositions of the particles. But the problem is, you may wonder, how do you validate, how do you calibrate your data, right? That's a good question, because when I started this, I'm thinking, because you have a different focuses, and also the samples could yield similar signals. How do you validate the different those particles of different colors correspond to what kind of polymers, basically? So you need to perform a very good calibration. Also, it requires a large data set processing using, special programming techniques. to analyze the compositional information. So again, it's not very straightforward. So the next one is fluorescence. Everybody is aware of fluorescent microscope. As long as the samples are fluorescent, you either label it with a fluorescent dyes, but the problem is you have to make sure the dyes will bend to the plastic particles specifically without the, staining other particles. And also the background interference is evident, especially for environmental matrix. Here are a few studies showing the enhanced contrast for when people use a special design of fluorescent microscope, they are able to increase the power. So this is a regular confocal microscope, but then this is I'm not sure, I'm not an expert in this, but they use a special techniques for when they perform the fluorescent microscope, so they're able to demonstrate the enhanced contrast for nanoplastics. But again, they are generating red fluorescent light, so you can see. But here, this is our results. We use a commercial plastic particle, polystyrene particle. They're made with a fluorescent dye inside their particles, not outside, not the surface coating, but inside. So then when we perform this study, we are not really, worried about the contamination or the coating detachment during the experiments. Okay, so besides visualization, again, you can see them, and the power, the ability that for us to differentiate the chemical composition is still limited, right? When you look at them, they're usually the studies usually employ a very single one or two types of plastic particle they can differentiate. But when you deal with the environmental samples, the samples could come in with different compositions. So here I want to just highlight a few common techniques that people have been using, like FTR, Raman. And you may wonder, so since they are very famous and also very powerful, why can't we just use them? So the problem is this, I have highlighted this, you should know that the Raman FTR would have a very limited spatial resolution. Spatial means how small you can see. So basically, the Raman, for a good Raman technique, spectrometry, you may, be able to detect the plastic particles, less than, around the 10 micrometers. So this is a huge, in our opinion, even though this is already small compared to the 5 millimeter plastic fragments, right? But again, in nanoscience, this is too large, right? And also there are emerging techniques called a micro Raman spectrometry or micro FTR. Again, the maximum or the best resolution that they can achieve is still not really below 1 micrometer detection limits. So anything below that, we're not going to see this very clearly. So as opposed to those regular Raman or IR, here I want to say there are a few techniques. For example, this is called a surface enhanced Raman spectrometry. Remember the previous slides I showed you that people use the silver nanowire as a filter to capture the plastic particle. That technique basically rely on so-called surface enhanced Raman spectrometry, where on your surface, on the substrate, you need to have a gold or silver elements to enhance Raman signal coming from your samples. For example, this is the image. This is a link to the study if you wonder the details. So what people have done here is on the substrate, which is a commercial Sears substrate, and they have a gold coating. And then on the surface of this substrate, we have PS and PMMA spheres that has very low diameter, 360 nanometers and 500 nanometers. They're really small. They're not, again, if you use a IR or Raman, you cannot see them. But on this design, you're able to see, Sears mapping. At least you can see something with the signals. Again, if you wonder why the signal is low, this is pretty much, related to the Raman principle. You can check the, the Raman principle why the signal is low when the sample sizes are small. Another powerful techniques here I want to demonstrate is called AF-IR. Again, if you look at the title, now we are not merely visualize materials. In the meantime, we're actually able to identify the chemical compositions or similar, or commonly called the chemical mapping. So when you map the samples, right, using AFM. Lots of people understand that AFM is basically a scanning probe microscope. You're using a probe, a very tiny AFM probe to scan over the sample surface. Then you generate morphological, images, right? As you can see their morphology. But again, in our research, we're not just, interested in the morphology. We also wonder what they are, right? So this is a problem for most of the AFM researchers. So if you combine AFM with IR, as you see from the image to your right, so you have a regular AFM configuration here. You have a cantilever, which respond to, the sample when the cantilever tip approaches the sample surface. And then the sample, the surface will certainly have interactions like a force of attraction or repulsive forces against the AFM cantilever. And then this kind of reflection or AFM tip vibration will be measured by the scattered laser beam. As you can see, there is a deflection laser, right? And then the deflected laser signal will be captured by a photodiode. So this is how AFM works. And in addition to the regular AFM, here there is a pulse tunable IR source. So basically, there is a IR laser source, like a laser pen. Basically, you shoot a laser, and certainly you can choose the laser wavelength, the light wavelength. You can choose a pulse signal, the frequency, something like that. So you can generate basically a pulse absorption of IR. So instead of measuring how that FTR or Raman rely on the signal, the IR signal absorbance or transmission, right? Depending on the thickness of sample. So in this AFM IR, we're not measuring any IR signals through transmission or absorbance. We are actually measuring indirectly. So this is a very smart idea. So once the sample, usually the polymer, polymeric samples, right? Once they absorb this IR energy, so the samples will expand. The sample, anything, will absorb energy. They're going to swell because of their, the expansion. This is called a thermal expansion. So when this laser is not a continuous supply, right? You can see this is a pulse. So you pulse it, and then the sample, like for example, plastic particles, they are going to swell and shrink, and perform such vibration. And this vibration magnitude is maximized when the IR frequency or wave number matched the actual IR wave number. I hope you understand that. When you use FTR, you scan the sample using a range of a wave number or frequency, right? So when you identify surface functional groups, you have a pretty large peaks, right? That indicates the maximum absorbance of IR signal, basically. So on this design, AFMR, it will generate a similar result or identical results, but not relying on the signal from the transmission or absorbance. Instead, it measures the expansion. The expansion is detected by the scattered laser beam. So this is the whole idea. So you can use this for a variety of different kinds of polymers, not merely the plastic particles. Actually, we have been using this for polymer membranes, like membrane filtration membranes. As you know, I'm a membrane researcher. So we are very interested in, for example, or not merely us, the industries, like membrane industries. They wonder when they fabricate a certain membrane, they coat with the membrane with additives, and they wonder where the additives are located, or what kind of chemical additives are out there, or what are their distribution on the membrane. So when you perform AFM mapping, you can see their morphology, no problem. Secondly, we also wonder here on this polymer matrix what they are. Are they polyamides, or they are polystyrene, where they're located. So we use this AFM and to detect their signals at different places. The red and blue dots indicate the locations where the tips are actually engaged with the sample surface. Then we can map or perform a so-called chemical mapping. And this gives a very good, good information about the chemical compositions of the matrix. So coming back to the plastic particles, we can certainly rely on the same principle and generate information of, plastic particle morphology as well as their chemical information, especially after, let's say, you mix up the plastic particles, or the plastic particle have a chemical changes, chemical degradation. Then you need you need a tool to accurately describe their surface states. Similarly, beside the AFM and IR, so we can certainly combine the use of AFM and Raman. Actually, this is even more advantages because you probably heard about the TB-enhanced Raman spectrometry, right? We just previously talked about the surface enhanced, where on the surface, the substrate surface, you put some gold or silver, right? The other way you can do it is you have a tip, right? The tip may not be, AFM tip, but it's general, tier is called a TD-Pyr tip. It's pretty large. So on the tip of the scanning tip, you can coat it with a platinum, gold, or silver, some noble metals. Once you use the tip to detect the sample, and also in the meantime, you shoot a Raman signal here, this laser beam, then you can have enhanced Raman signal for some reason. This is called the tip-enhanced Raman phenomenon. So on the AFM, you have the tip over there, right? Very small AFM conductive tip, and you can place a Raman usually from the bottom. This is a commercial AFM Raman. They are very powerful. I have a study a few years ago. We used these techniques to detect the graphene, not the plastic particles. The graphenes, basically, the graphene absorb Raman pretty strong, very sensitively. And then we use AFM to analyze the surface, mapping, so look at the whole surface. And then in the meantime, the Raman signals will tell us where the graphene oxides or graphenes are located on the sample surface. So this synergy would allow us to merely visualize, but also chemically identify the particles. And so there are a very limited, in my understanding, I'm not sure why, but there are very limited study using these techniques. And following that, I also want to introduce a technique called the carbon force microscope. It's one of the modes of AFM. It doesn't require complicated, configurations with the IR or Raman sources. You do not need to. So it's just some operation modes. You can, so in our previous, many years ago when I was a student, we use this techniques to quantify, to map the quantum dots and nanoparticles, as well as even the DNA polymers. DNA is a polymer. So they generate different type of contact potential difference. So this is related to the work function difference between your tip and the sample. And you understand that your sample is always different from your tip, which is usually platinum tip, AFM tip. So once you generate the tip, work function difference, you can differentiate the materials based on the contact potential tip. So this is another way to not merely visualize, but also tell you what they are, right? When you look at this polymer, how do you know these polymers are DNA versus other polymers? You can quantify the work functions in a confined or in a well-defined environments. But unfortunately, the problem is, you can see we do have some polymeric studies using AFM. One problem is the sample material must be very clean. It should be free of surface contaminations. Carbon force microscope is very sensitive to surface states. What does it mean by surface state? For example, moisture. If you have a high moisture in the environment, ambient environments, moisture or water molecules would interfere the work function very, very strongly. So that's why I don't see many studies, especially for environmental samples. I never see a study utilizing KPFM to quantify or map the particles of plastic particles. But I urge everybody to think about it. Someday I'm sure this technique would be very useful. So besides the AFM, IR, Raman, and also KPFM, this is I would say most fancy techniques I've ever seen. This is very difficult to operate, but it's very fancy and powerful. So as you probably know, this is called a scanning electron microscope, where you have a counter, working electron reference. This is basically electrochemistry design. And then you have a tip, which is a working electrode, and you scan the surface, right? Then you can map the tip sample currents. And when, due to this tip sample current, you can tell where the surfaces are reactive or more reactive versus the other areas. And so AFM, AFM now. So then we can combine them together. Okay, we can perform AFM mapping. In the meantime, we can generate signals of a tip sample currents. So that why we do this? Again, we want to analyze local in situ surface states. So if you deal with the nanoplastics, again, how do you differentiate the nanoparticle, nanoplastic from the matrix? This is one of the techniques that are potentially useful. For example, this is some of the literature data, even though they're not directly related to polymers, but they are useful. If you look at the data, they use here, for example, noble metal particles. Again, not merely looking at the particle shape and size, but also we can tell their reactivity. Some of the particles might be more reactive. So if the surface, the tip currents, you can see the tip currents is higher or lower depending on how you run this electrochemical measurements. So then you can largely differentiate the samples from somewhere else. So you may wonder, you already differentiate based on the morphological images, right? This is true. But again, if you have a mixture of particles, for example, silver, gold, and plastic particle all together, can you really tell? Certainly not. You cannot tell what they are. So then at that situation, you certainly can apply the currents on the sample or tip to enable reactions with your matrix, your solution matrix. For example, this on the right bottom image that shows you a very good example where you have a catalyst right here. This is a catalyst, the platinum nanoparticles or other kind of particle, And then in the matrix, you have electrolytes that would react, I guess here, with the facility here. If you apply a voltage onto the sample would react with oxygen and produce hydrogen peroxide. And that hydrogen peroxide will migrate to the tip and react with the tip and generate tip currents, right? This is the idea. This is one of the design that you can use. So based on this design, see here, we can generate so-called all oxygen reduction reaction mapping and generate, depending on where you measure the currents, right? You can sense where the reaction sites are located. And so by, explaining such a principle, you probably realize that for plastic particle, this is a problem. So you have to make sure the plastic samples are electrically active or reactive. They must be somehow reactive towards the electrolytes, or otherwise you can still map them, but based on let's say the plastic particle interfere the design electrochemical reaction on your tip. That's also durable, right? So for example, your sample matrix is very reactive. You can generate pretty high currents. I think this is the example here. You can have pretty, so on the opposite side, so you can have pretty high currents on your matrix, on your substrate surface, but once your tip engaged with your sample, you have a limited or reduced tip current. That's the opposite methods to identify the inner plastic particles. That's I think that's also feasible. And also you need to have a very clean substrate as well as a clean matrix, to eliminate interferences for reactions. And also the entire operation is very laborious and costly, and even more costly than running SEM-TEM. But it's a good techniques for enable nanoscale characterization. Here, this is probably the last example. I'm not sure if you ever heard about that. This is called a photo-induced force microscope. This is also conducted on the AFM. Okay, so it's another one is a photo-induced force infrared spectrometry. They are all pretty much like AFM-IR, but AFM-IR detects the thermal expansion, right? It's the pulse expansion. And here in this application of AFM, instead of measuring the expansion, it's measured the tip sample forces. And when the sample, let's say the plastic sample, this is the demonstration of nanoplastics, PMMA, PDFE, PS. So when they exposed to the IR signal, the sample not merely expand, but also generate the surface molecules of the matrix, the polymer matrix. So it's going to shift in between the polarity, becomes more polarized versus less polarized. So this polarized states will induce a strong interactions. It's very complicated. I have no idea how. So the force, the tip and sample would have a very complicated, polar to polar interactions. So this kind of force will be recorded by AFM and then give you a signal here. As long as I see the signal similar to IR over different wave number, you're going to generate a so-called signal. So this signal is not the magnitude of the expansion, but it is actually expressed as a photo-induced force in millivolts. So it's pretty much, the scatter is probably measuring the scattered laser signal. I certainly, understand there are a lot of challenges besides what I just covered from pre-treatment separation as well as visualization, quantification. When we speak of quantification, chemical identifications, there are a lot of techniques out there, like pretty much based on the mass spectrometry, depending on how you hybridize with or hyphenate your techniques using thermal desorptions or other means. Laser, for example, you can use a laser desorption techniques. But I don't have time to go over everything. But I want to just say that the challenges are obvious. For example, when you deal with a small sample size, small nanoplastic particles, they are very, very small, right, in size, in mass. So then when we try to detect the polymer vapors, when we heat them up, usually that's the problem. We do not have, for example, 100, 200 micrograms. So then that means you're not going to have a pretty good signal using these techniques, right? So that's pretty forward. So with that, I want to just jump to my major conclusions. I think similar to nanoplastics, nanoparticles, we have been facing similar challenges for nanoplastics, special nanoplastics. I basically overlooked the microplastics since that's not the focus of my talk. And then the plastic particles could decay or damage during the sample pre-treatment, even during the sample analysis. If you think about the electron microscope, if some of the polymers are thermally vulnerable or very sensitive, they may melt down at the high elevated temperatures, right? So like a PE, they may melt down, they may change their they may deform or change their phase. So yeah, so when you perform SEM, TM, you must be careful. Interferences from matrix, proteins coexisting, nanoparticles could render signals IR, Raman fluorescence that largely prevent us from accurate quantification sometimes. And the AFM techniques, even though very powerful, as you see, very fancy, showing us a very diversity of functionality. But the thing is, they are constrained, I would say, to laboratory conditions. When you deal with the environmental samples, it's really, really hard to operate. Okay, so with that, I'd like to conclude my presentation. Thank you so much for your attention. I'm going to be open for questions. Thank you. Let's see here.
Awesome, thank you, Win. And yeah, like you said, if you have any questions, you can put them into the Q&A section, the tab. We do have a little time for a couple questions. So we do have one from Lindwo. She says, thanks for the talk. Is there any technique to differentiate the plastic type? For example, PP, PE, etc.
Yes, of course. The regular way to differentiate the bulk polymer, you didn't really, I guess you're talking about the large bulk plastics, not merely the nanoplastics. Usually we're going to just use FTIR Raman to quickly, based on, they all have existing database for the signal library, right? So based on the library, we're able to really differentiate those commonly used plastics, types. But again, remember my presentation also mentioned that when you deal with a small size of the plastics, then those techniques are not going to be very useful because of the signal, the detection limits. Okay.
Okay, next question. One of the difficulties we commonly encounter in micro nanoplastic analysis is limitations to surface area mapped. How much of a surface area can the AFM techniques discussed realistically cover? 20 centimeters, 10 centimeters, more or less? Okay,
that's a very good question. Yes, for those people, for those who are not familiar with the AFM mapping, yeah, so usually they have, they're puzzled about the scanning surface of the, when you try to quantify the plastic numbers for surface, certain area of the entire sample. So unfortunately, this is one of the major disadvantages of performing AFM versus, versus other tools such as, fluorescent or electron microscope. You can scan pretty large. How large? Usually, I would say a few hundred micrometers all the way, right? You can see large areas. And then for AFM, to answer your question, for AFM, I would really recommend the small areas. For for nanoplastic, right? So small areas are usually 500 nanometers by 500 nanometers. Sometimes, if you want to analyze microplastic, which is fine, which is visible, then you have to increase your mapping area. But the maximum, I would say the maximum that AFM instruments could allow you to map is somewhere between, I would say 5 micrometers, maybe 10 micrometers maximum. When you increase the mapping area, you're going to increase your time, your mapping time. Mapping time could range from a few minutes all the way to 30 minutes. So the longer you map, of course, your throughput rate is low. So also you have challenges of damage your tape for large mapping sweeping area, because you map so much area, then the tips, AFM tips could be damaged. Yeah.
Awesome. Do nanoplastics bioaccumulate in nature and cause adverse effects? For example, hormone changes, sex changes, cancer, etc.
Okay, again, I'm an environmental engineer. I know this presentation is recorded, and I cannot really endorse all the suspects from the public, and certainly to be serious and also be responsible. I want to say that those kind of negative impacts on human health or the ecological system are possible, largely possible. The reason I said that is bioaccumulation of nanoparticles is already proven, in the past few years. So nanoplastics have no reason not to show such bioaccumulation, right? Secondly, the plastic particles contain, as I mentioned, they contain a very trace level chemical additives. Those additives could be, causing hormone, disruptions or endocrine disruption effects. So you never know. I would say we should be very conservative and prudent when we deal with this new emerging contaminants. Yeah.
Awesome. I think we have time for, this will be the last question. Are all cosmetic microplastics outlawed now, and is it being enforced?
Yeah, so I remember it's probably in 2018 or 2019 in New Jersey, at least the law, there's a law that already banned the use of plastic beads in cosmetic products. So now you're not going to see plastic beads anymore in the rinsing foam or something, because the plastic beads are one of the dominant species detected in the marine environments. But you still see the beads, right? The beads is made of other polymers or any other thing that are not as harmful or as persistent as the plastic particles. Yeah, so we're not really worrying about that anymore, at least in New Jersey. I'm not sure about other states or other countries. Yeah.
Okay, awesome. Well, yeah, that's going to be it for today. So once again, I'd like to thank Dr. Zhang for participating, and thank you all for coming. And that's it.
Yeah, thank you all for your time. Keep in touch. Bye.